We are on chapter 4, section 3, and we're really going to talk about how proteins fold. And there are some big general principles of quaternary structure. So the fourth, quaternary. And we know already that the primary amino acid sequence determines our three-dimensional structure, and therefore our three-dimensional structure defines our biochemical function. So all of that has to do with the primary structure. But how is it that you can have a primary structure and turn that into folding so that we get a stable three-dimensional structure? And one of the things that you have to really remember when we talk about these general principles is that molecules are really flexible. They're constantly moving, they're constantly fluctuating. They are not stagnant. And so because they're flexible, it allows the proteins to basically try on or test different three-dimensional structures during the folding process. So they can kind of attempt and see what works. And you really also have to remember that proteins are always in contact with some kind of a solvent. And that solvent has a major impact on folding and therefore the eventual three-dimensional structure. And so, right, if you're a cytosolic protein, your solvent is water. And so you're going to make hydrogen bonding with water, and that is going to drive the hydrophobic residues into the inner pocket of the proteins and help to fold. And then you got to think about membranous proteins. That's going to be flip-flopped, right? It's going to be better for them to have those hydrophobic residues on the outside instead of the inside. So what are our general principles? I feel like I got already off on a tangent. Our primary sequence leads to a limited number of folding pathways. So there was a really cool thought experiment. So if protein folding is 100% random, if it's all random, right? And you mathematically calculated how long it would take for an amino acid sequence to fold into all the possible arrangements, you can't calculate that. It's too big. So one guy said, okay, well, what if, right, we had an 100 amino acid protein sequence, and each amino acid tried 3 possible phi and psi angles, and that's it. So just 100 amino acids and 3 possible phi and psi angles for every amino acid. And if it were 100% random, it would take billions of years to try all of those possibilities out. Well, protein folding occurs on millisecond scale, right? So that's not going to happen. That would take too long. So there are only a limited number of possible pathways. And so it restricts, you don't have to go through all the possibilities because some of them are non-viable. Then you have to remember that there must be a free energy difference between the folded and the unfolded state such that being folded is favorable, okay? So remember that it depends on enthalpy and entropy. And so our entropy, right, has to do with a lot of times with water, right? So think about if water is surrounding this protein that's folding, right? The more that those hydrophobic residues turn inward and get away from the water, right? This is the hydrophobic effect that we talked about before, that you have a decrease in the amount of order that the water molecules have to be in. So you actually have an increase in disorder. And so that really helps to help the free energy of that state, right? And so you have kind of an equilibrium where you have folded and unfolded, you know, and the protein can go back and forth. But really what happens is that equilibrium will shift towards the folded state for most of the proteins due to those favorable conditions like delta S, right? And the other thing is, my iPad went to sleep. I talked so long that my iPad went to sleep. That's really funny. And now it won't recognize my face. Okay. So the third part of the general principles that coordinate folding is that in vivo folding is very different than in vitro. Remember, in vitro is in the test tube, right? So when we study all this folding in a test tube and all that kind of stuff, it's not really what's going on in the cell. And usually when we eat proteins, we don't go over about 10 micromolar. And the reason that we don't go over 10 micromolar is because if we had that much protein inside of our test tube, that protein would aggregate. And you would have like hydrophobic regions of the protein interacting with each other and forming aggregation. But because of the cellular environment, usually protein concentrations are about 30 times higher than what we do in vitro. And so the environment is really different in the cell than it is in the test tube. And then you also have to think about how proteins actually fold in the cell. It's not, when we're in a test tube, basically what we have is a synthesized complete protein. It's the whole thing. It's all the amino acids. And then we try to get that whole thing to fold. But that's not what happens in the cell. As the protein's being made, little pieces of it can start to fold. And so it doesn't all have to happen at once. And so you have to remember that that's very, very different between in vivo and in vitro. All right, so how is it that the cell produces so many properly folded proteins? And so really the molecular basis for this high fidelity folding and refolding, because a lot of proteins are misfolded and then have to be refolded, is due to these special helper proteins that we call chaperones. And the chaperones provide basically favorable interaction surfaces. And those interaction surfaces will facilitate the formation of stable three-dimensional shapes. And so they'll help newly synthesized proteins fold properly. Then once we have proteins made, sometimes they fold improperly. And so they can go in and pick those up and rescue them and then refold them. And then they can also help to disrupt protein aggregates. And we won't talk so much about this last one, but we're going to talk about the first two. So I want to give you some examples of the two, and your book says the two types, but it's really the two most common types. There are other types of chaperone proteins. There is the clamp type and the chamber type. And the clamp type, we're going to watch an animation in a few minutes about these two different types of chaperone proteins. But the most common or well-studied clamp type chaperone protein is called the heat shock protein, or HSP. And the one you'll watch a video on is HSP1. But what happens is cells can undergo cellular stress of high temperature, right? And if that happens, most of the proteins are going to denature. And so these chaperone proteins, these heat shock chaperone proteins, will provide a way for the cell to basically recover from high temperature extremes by refolding the proteins when the temperature returns to normal. So it can kind of like wait it out. And what you'll see is the heat shock protein is expressed in all cells under all growing conditions. And they're actually required for protein folding even in non heat shock or heat treated cells. So I think that is HSP 70. Is that what we're going to watch? I don't remember which number. There are quite a few of them. Anyway, so the heat shock protein chaperones function in folding of normal proteins as they're being synthesized and in this like rescue recovery thing. And so it's kind of a clamp. So it kind of like grabs the protein to be folded, helps it, and then releases it. And the other is a chamber type, and you think about a chamber, right? And so the proteins have to enter into the chamber, are facilitated, folding is facilitated, and then they're released. And both of them require ATP in order to facilitate the folding. That's an energy dependent reaction, and so it needs ATP to complete it. So when we think about protein folding, before we watch the video on how it actually happens, I want you to understand that protein folding is cooperative. So remember when we talked about DNA and how when you looked at the melting temperature of DNA, and when a DNA strand starts to unwind, it's cooperative, and then it like completely unwinds, and vice versa. Well, protein folding is the same thing. You can actually monitor how much of a protein is folded and how it's folded using something called circular dichroism. Circular dichroism, or CD, they call it. And so circular dichroism measures the difference in absorption between right-handed and left-handed circularly polarized light. But you pick different wavelengths that are between like 190 and 250 nanometers. And what you can do is, looking at how much light is absorbed, you can identify, oh, we have this percent alpha helices, we have this percent beta sheet, we have this percent unstructured region. And so knowing that, you can actually take that reading, right? And then you can increase the temperature, and you can measure the percent of the protein that's folded. And then as you increase the temperature, it's going to become unfolded. And so that's exactly what this curve is. And you can get a TM, or a melting temperature, for your proteins using this circular dichroism. And what you'll notice is that this is a really, really, really steep part of the curve. Well, what does that mean? That means that it's cooperative in transitioning from folded to unfolded state. So it happens very, very rapidly. So you have this kind of equilibrium between the folded state, the folded state and the unfolded state. And it transitions back and forth very, very quickly. So how do they actually fold? So before we talk about chaperones, let's talk about how proteins actually fold. And so there are 3 proposed models for protein folding. And we're going to talk about each one, but what I want you to understand before we talk about each one is that for any given protein, it could be using one or more of these folding mechanisms to make that final folded state. So it's not just one. It's usually going to be a combination, and we're, as much as we wish we understood this completely, we don't. So the first one is called the hydrophobic collapse model. And in the hydrophobic collapse model, it actually predicts that there's a clustering of the hydrophobic side chains. So clustering of hydrophobic side chains, right? And that is what drives the formation of secondary and tertiary structure, right? And so once you get secondary, then you get tertiary, then you have this completely folded structure, right? So that's kind of saying that it's really the hydrophobic interactions folding in that make the protein fold properly. The second one is called the framework model. And in the framework model, you're predicting that first you're going to make secondary structures. So you make secondary structures, right? And then you make tertiary structures. And so it's that secondary actually forming before the tertiary. In the hydrophobic collapse model, kind of you're driving secondary and tertiary all at the same time, right? In order to get that quaternary structure. But in the framework model, it's kind of like a stepwise. Okay, I'm going to make secondary structure. That secondary structure is going to drive my tertiary structure. So it's kind of like interdependent. And then the last one is called the nucleation model. In the nucleation model, it's similar. We're saying we make secondary structures, then we make tertiary structures, but we're saying that the tertiary structures are driven by the localized interactions between the secondary structures. So driven by localized interactions between secondary structures. So in the framework model, it happens sequentially, but they're independent. In the nucleation, it happens sequentially, and tertiary is dependent upon interactions between secondary. So it's 3 different models, and we were that all proteins kind of undergo a combination of these, with, of course, exceptions. All right, so I want to actually show you how some of these chaperone proteins interact. And so we're going to watch a little video.
In order to be functionally active, proteins must fold to give rise to a specific 3-dimensional structure. Although many cellular proteins are thought to fold successfully on their own in the cytosol, some proteins appear to require the assistance of special proteins known as chaperones to complete the folding process. One of the best characterized chaperone proteins is heat shock protein 70, or HSP70. HSP70 is a clamp-like protein that, when bound to ATP, undergoes a conformational change that reveals hydrophobic groups, allowing unfolded or misfolded proteins with exposed hydrophobic residues to bind. When ATP is hydrolyzed, HSP70 closes on the misfolded protein. After ADP is released, HSP70 opens to free the partially refolded protein. This cycle can be continued until the protein is completely folded. The GRO-EL-GRO-ES complex is an example of a bacterial chamber-like chaperone. The GRO-EL subunit consists of 14 identical subunits arranged in 2 rings of 7 subunits each. Each GRO-EL subunit has 3 protein domains: a substrate binding domain, an intermediate domain, and an ATP binding domain. The substrate binding and intermediate domains form the walls of the chambers, while the ATP binding domain separates the upper and lower chambers. GRO-ES also forms a 7 subunit complex and functions as a cap for the complex. The GRO-EL-GRO-ES protein folding cycle consists of 4 steps. First, GRO-ES and 7 ATPs bind to the upper GRO-EL ring and trap an unfolded protein inside the chamber. Then, in the lower chamber, a conformational change occurs that releases GRO-ES, 7 ADPs, and a partially refolded protein. After a few seconds, the 7 ATPs in the upper chamber are hydrolyzed, causing a conformational change in the upper chamber that facilitates protein folding. Meanwhile, the lower chamber accepts a new unfolded protein. In the last step, 7 ADPs, GRO-ES, and the partially folded protein are released from the upper chamber, and the cycle repeats itself. An estimated 30% of protein synthesized in the cell must be refolded or degraded. If left to accumulate, misfolded proteins can aggregate, interfering with cellular processes and potentially leading to disease.
Okay, so let's talk about that, right? In the video, she just said that an estimated 30% of proteins that are synthesized in the cell need to be refolded or degraded to prevent accumulation. So what happens if they aren't? Well, you get some diseases, right? So there are 2 different basic categories of what happens in folding diseases, right? And so they can be due to 1 of 2 things. They can be due to a mutation in the protein coding sequence, right? And that's in the gene, right? So you have the mutation in the gene that leads to a difference in the protein sequence. Or 2, you can have an accumulation, an accumulation of misfolded proteins without any amino acid changes. Now that's really cool, and we're going to talk about each of them. All right. So if you have a mutation in the protein and it changes the amino acid sequence, you can guess that if it causes a disease, then that protein is no longer going to function the way that it was supposed to, right? You could have, we talked about the different kinds of mutations, right? We talked about you can have an insertion or a deletion. You could talk about where you have a frame shift mutation, right? All the different things that happen. And so if you have that amino acid sequence and it becomes mutant, there are two things that can happen, right? So any of these disorders listed, we have an amino acid sequence that is mutated. And so a lot of times what happens is that because it is mutated, then that protein becomes degraded. And so you end up with a loss of function, right? You could have a situation where you have a mutant, right? But then the protein does not get degraded, but it builds up. So because it builds up, it aggregates within the cell. And so that can lead to a gain of function. And then the third type is that and that's not actually true. Oh, yeah, okay. So you can have a normal, so here's another mutant, right? So the third type though, so you have 1, 2, and 3, is that you have a normal sequence. So you have a normal gene, you have a normal amino acid sequence, and regardless of that, it still ends up aggregating. So you still accumulate, it misfolds for whatever reason, and it aggregates. And so regardless of whether there is a mutation or not, if it aggregates, we call that a gain of function mutation. So I think this stuff is super cool, like everything else in biochemistry. And so we're going to talk about the 3 different examples of the different misfolding leading to disease, right? So the first one is cystic fibrosis. So in cystic fibrosis, you have a gene and it contains a mutation. And so that mutation leads to a misfolded protein. The cell recognizes this misfolded protein and says, that's not good, I need to get rid of it. And normally that's a good thing. But in cases where so much of the protein is degraded that you don't have enough left to do normal cellular functions, it becomes a problem. So in cystic fibrosis, the gene actually encodes for a chloride ion channel. Chloride ion channel inside of a lung epithelial tissue. And so you get this mutation, the protein misfolds, the protein is degraded. That means that greater than 90% of the protein, of that newly synthesized protein, gets degraded before it can be inserted into the membrane. So now you can't pump chloride ions across your membrane. If you have an imbalance in ion concentration, guess what's going to happen? You're going to lead to fluid accumulation in the lungs. And if you have fluid accumulating in your lungs, you're going to have pulmonary dysfunction, and you'll end up with infections and difficulty breathing and lots of serious, serious problems. All right, the second one is Huntington's, right? Huntington's is an example of a mutation where we have an expansion of a polyglutamine. So you actually have it CAG, that's a C, that's supposed to be a C. CAG, and CAG codes for glutamine. And so this CAG triplet gets repeated over and over and over and over again. And if you remember your structure for glutamine, let's see if I can kind of zoom in and get myself some space, and we're going to draw it, right? So there's our alpha carbon. We have this, we have this, right? Okay. No, let's, no. Okay, so then we have here CH2, then we have another CH2, then we have CH. Now, what's hanging out here, right, are lots and lots and lots of possible hydrogen bonding in both parts of our side chain, right? So if these things can participate in hydrogen bonding, and you have duplicated them over and over and over and over again, what's going to happen is that protein is going to misfold, right? You have that many glutamines one after another. It's going to hydrogen bond with itself. It's going to hydrogen bond with other mutated Huntington proteins, and that's going to lead to aggregation. It leads to so much aggregation that you end up with neuronal cell death. Your neuronal cells start dying. Now you have a neurodegenerative disease. So that's Huntington's disease in like 30 seconds. Creutzfeldt-Jakob disease. This is actually an example of no change in the protein coding sequence. And it's really, really pretty cool. It's not the only one. Alzheimer's is another one where we don't have a mutation. We have a normal protein product in Alzheimer's, and the normal protein product is cleaved. It produces a hydrophobic protein fragment. That hydrophobic protein fragment aggregates, and then it creates problems. But if we go back to Creutzfeldt-Jakob, it's an example of a transmissible spongiform encephalopathy. So a TSE, you've probably heard of it because you know about prions, right? So a prion protein or a PRP, right? Scrapie in sheep, mad cow, kuru, right? And kuru, Creutzfeldt-Jakob is an example of kuru. It's kuru in humans. And so in humans, you have this normal PRP in cells, and we call it PRPC, right? For normal cellular prion protein. And we think it functions as a cell surface glycoprotein, so it kind of sits on the outside of the cell. But we think that the normal one gets misfolded and turns into PRPSC for Scrapie. It's an abbreviation for Scrapie. So it's a misfolded form. And this new misfolded form aggregates. And because it aggregates, right, then it's going to interfere with normal cellular processes. And we don't have all the answers. We don't understand why it aggregates. It's really, really poorly understood, but we know it does happen. Okay. And like, I wish I could talk about that for like a really long time, but I think that's all I got. Okay. And that's it. The end. Yay. End of chapter 4. Okay. Y'all have a great day, and I will talk to you later.